Are you feeling better about the world today?
A little bit. If I could get this to work, that'd be even better. How's it going, Win?
Better today.
Yep. What's wrong with that?
Tom's throwing his computer around.
I threw my computer around. It was great. I was feeling really good. So, B, I got to run and get the light in the lab.
Okay.
So if this presenter comes on, would you say hello to her and make her a co-host?
Yes, we'll do. Also, it didn't say it was recording yet.
Yeah, it's not. So if you would record it, that'd be great.
If I could record it?
Yeah. Do you have enough space on your computer?
I think so.
Okay. Thank you.
Yeah, no problem.
Be right back.
There are some aspects of doing this by Zoom that I like. One is I can eat my dinner while we're doing it. But I do miss standing around and chatting with everyone.
So true.
Yeah. It's very different on here.
I have my seat, and I'm used to sitting in my seat, and now I can't do it. I have to sit here in a chair in my kitchen. That's about it. That's no fun. Better than nothing.
That's true.
I envy you in Paris, Larry, even if it's only virtually.
Well, I went all day watching the inauguration stuff, so I'm tired.
Me too, and I enjoyed very much being able to see all the different parts of it that if I'd been there in person, I wouldn't have been able to see.
Oh, yeah, right.
I particularly enjoyed the poet laureates
Holy
presentation.
Yes.
Holy moly.
Yeah.
Only
And
two-
having the pledge said with ASL.
Yeah, that's right. Yeah, the fireman, right? He was a fireman.
Mm-hmm.
Fire was a female firefighter.
Yeah, firefighter.
That 22-year-old poet laureate has more wisdom than I do.
Well, to come up with such a good poem in such a short period of time, you know,
She
really
nailed
speaks
the
to her
whole
talent.
insurrection thing 2 weeks ago too.
Yeah.
She addressed that more head on than anybody.
Yeah, I was reading a little bit about her. She started writing poems when she was 3.
Wow.
She wasn't potty trained, I don't think, but I don't know.
Oh, she probably was. She's a girl. They potty train about half a year younger than boys.
Really? I didn't know. I didn't realize that.
Yeah.
I'm still not potty trained, especially I've gotten older.
Well, I was going to say, we get over it as we get older.
Yeah, you know about that, Tom, don't you?
I tell you, it's good stuff. Hi, Eileen. How you doing, Sandy? How's it going, Tom?
Okay,
How's the flying squirrels?
they're doing just fine.
How's their feeder doing?
Constantly getting filled.
No. The feeder-
No, I know what you're
Yeah.
saying.
Okay.
Not so good.
Bob. Good. We have a speaker. Oh, there she is. Hello.
Oh, can you hear me?
Yes, we can.
Okay.
Hello.
Wait.
We're about to make you
There was something wrong with my AirPods. Can you hear me now? Yeah, okay. I can hear you.
Okay, and you're now a co-host?
Okay. Do you want me to try to share my screen? Yeah.
Correct. Yes, please.
Okay, good. Okay. Yes, this looks good. And it will
Good.
actually look like this.
Excellent.
Yeah.
And then do you have any videos that you want to test drive? Or is
I don't.
Good. Thank
I
you.
don't have any videos, no.
And then Claudia, could you do me the favor of pronouncing your first and last name for me?
Yeah, I mean, it's very robust to however you want to pronounce it. That's Claudia Solis Limus.
Solis Limus.
Yeah.
Solis Limus. Okay. If I don't nail it, go ahead.
No,
and,
that's great. No.
well, I can do it now, but in 5 minutes.
Yeah, that's okay.
And
Yeah.
then I will ask you the standard 5 questions. Where were you born? Where'd you go to the equivalent of high school? Where'd you go for your undergrad? Where'd you go for your master's or PhD? Any postdocs? And then when you arrived here.
Okay, and I do that at the very beginning, right?
Yeah, well, I'll just ask you once we start on the formal.
Okay.
Good.
Yeah.
Thank
Sounds
you.
good. No, thank you.
Mhm.
This is my first time here.
Good. Thanks for coming, Eileen.
My neighbor, Michael Rip, told me about this. It sounds like
Good.
very interesting. What is the topic tonight? I didn't look it up.
Claudia, you want to tell her?
Yeah, so hi, Aileen, is that your name? Yeah.
Eileen.
Eileen. Hi, Eileen. So I'm Claudia. I'm an assistant professor at UW-Madison. I'm on the Department of Plant Pathology, but I'm a statistician. So I'm going to be talking about data science and how we analyze data in this big data era.
Okay, sounds interesting. Thank you.
Yay. All right.
Good evening, Tom and everyone.
Good evening.
How's it going, Kathy?
Good evening.
Tell you.
Are you watching on two screens because you're so interested tonight, Tom?
I can't figure out how to run the thing.
Sounds like most people.
Every week is a new adventure in how to run two screens at the same time. I know. I'll just try to figure out how to. Oops. I don't want to click out of the other one because I'm afraid that I'll shut everything down. All right. I think I'm down to one.
You're still showing up twice on my list.
Okay, I'll try this.
Twice on mine too.
Okay.
Not that we're not glad to have you.
I am. Wow. Yeah, I got to figure out. So there's me. I'm a host. Yeah, it's 7 o'clock. So
Why worry about it?
I'm not going to worry about it. I put a sheet of paper over my other lens, so hopefully that'll stop it. One never knows. All right. Well, welcome everybody. I'm going to start going through the lineup for next week. If I can get this to open back up. Come on. There it is. Next week, on January 27th, Sean Carroll, who's now with the Howard Hughes Medical Institute and has been for actually many years. He's the vice president of science education there and is a president or a professor emeritus from the Laboratory of Genetics here. Will be zooming to us from Maryland on Eastern time, so he'll be coming in about 7:45 and we'll be starting at 6:45. He's going to speak to us about his latest book, which is entitled A Series of Fortunate Events, Chance and the Making of the Planet, Life and You. What we're going to do is roll a presentation that he has sent to us. That way we don't have to worry so much about the YouTube connection, not the YouTube, the Wi-Fi connection. And he'll be here live at the intro. We'll roll the presentation and then we'll go back to live and hear some comments and do questions and answers. On February 3rd, Andrea Strzelik, who's with the Engineering and Engine Systems program here, is going to talk to us about the future of hybrid vehicles, electric versus electrified powertrains. It's a pretty great talk. I just heard her give it yesterday, and I'm delighted that she's going to be able to come on live on February 3rd. February 10th, Mariah Zapara from Penn State is going to be our special guest as part of Darwin Days. We get to do this every year in conjunction with the organizers of the UW-Madison Darwin Days. And she's going to be speaking about her work in neurovirology the genomics of pathogen variation and neuron-virus relationships. So that ought to be pretty cool stuff. And then on February 17th, we're going to have a panel of 4 evolutionists talking to us about human microbiome interactions. So get to have a couple of evolution talks as part of Darwin Days. So with that, why don't I start in with a more formal intro and you can start up. Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension in Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night by Zoom, 50 times a year. Tonight, it's my pleasure to introduce to you Claudia Solis-Lemus. And she's an assistant professor here at UW-Madison in both the Wisconsin Institute for Discovery and the Department of Plant Pathology. Claudia, how are you doing?
I'm fine. Thank you very much for the invitation.
Oh, and just as a reminder, folks, Claudia was all queued up to give the talk last March 11th. And that was the day that the hammer came down and everything got shut down. And so here we are 10 months later. I really appreciate you hanging in there with us, Claudia, and it's a delight to have you here. We'll go through the 5 questions. Where were you born?
I was born in Mexico City.
And where'd you go to high school?
In Mexico City as well.
Wow.
Yeah.
And where'd you go for your undergrad and what did you study?
Yes, so that was easy as well. I was in Mexico City. So I went to high school, undergraduate in Mexico City, and I studied applied mathematics and actuarial sciences.
Excellent. And then where'd you go for your master's or PhD or both?
Both I did them here actually at UW-Madison. I did a master's in mathematics and a PhD in statistics.
So did you actually go to live in Van Vleck Hall then?
Well, what do you mean by live? Like, yeah, I was in the building.
You're one of our first speakers that would be familiar with Van Vleck. That's
yes,
great.
I am in fact, yeah, familiar with Van Vleck, yes.
Good. And then you did a postdoc?
Yes, that's correct. So I did a short postdoc here at UW once I finished my PhD. And then I went to Emory University in Atlanta, where I did another postdoc before coming back.
Good. When did you come back here?
2019, so fall 2019.
Very good.
Yeah.
I'm looking forward to your talk. You're going to be talking to us about Through the Looking Glass of Data Science. Everyone, would you please join me in welcoming Claudia Solis-Lemus to Wednesday Night at the Lab. Claudia.
Thank you. Thank you again for the invitation. I'm really excited to be here. And yeah, like you said, my talk was scheduled for March 11, but that's what the day that the campus lockdown started. So we had to cancel everything. But here I am. I'm happy to be here. It would have been obviously better in person. We all miss person communication, but I mean, virtual talks are good too. So I'm going to be talking about data science. Like I said, I am in the Department of Plant Pathology, but my background is statistics. I'm a statistician. So when I tell people that I am a statistician, sometimes what they think that I do is that biologists give me their data, and then I put the data in the computer. I press a magic button in the computer, and then voila, the data is analyzed. So it's just like magic. And then I'm just resting the rest of the time. The computer is doing all the work. Of course, the reality is a bit more complicated than that. And we're going to guide you through today is that I see data science as the combination of 3 major areas. So there is the domain expertise that has to do with biology or the data that you are analyzing. There's mathematical or statistical theory, and there's also computer science to actually program the computer. And I'm going to start with a very simple example to illustrate these 3 parts of data science. So let's pretend that we want to classify frogs from lizards in this slide. And as humans, we can all do this, like even a kid can do this. And this is actually a picture from a collaborator whose slides I'm stealing right now, Daniel Pimentel. He's a faculty here at UW. So that's a picture of him as a kid. And that is the homework. He has to separate frogs from lizards. And like I said, this is very easy task for humans because we can distinguish that. But how do we train a computer to make this distinction? That is the question. And the computer, we need to give information to the computer that the computer can understand. And one of the key things that we can say as an expert on lizards is that it seems that lizards are longer, like the ratio of length versus width is higher on lizards compared to frogs. This is just one random fact that I'm saying, but this is the one I'm going to use for the example. So if we plot all of our data into a two-dimensional plane where we have the length, we measure all of our lizards and frogs, and we plot the length on the x-axis and also the weight in the y-axis, we can substitute each of our organisms or each of our animals in the sample by a point in this two-dimensional plane. So I'm going from the physical world where I see the lizard, I see the frog, to a mathematical world where I only care about the measurements. All that I care about these animals is the length and the weight, and that's represented by these two coordinates. So every organism in my sample, I can actually move it to this mathematical space by just one point in this plane. And then the question is there such a line or such a structure that separates the points that correspond to frogs to the points that corresponds to lizards? And if I can find this line, then any new point that I get, I would be able to know without looking at the animal, whether it is a frog or if it's a lizard. So here is where the statistical and mathematical expertise comes into play, because that's finding the line. And as you probably remember from your math classes, just getting this line is just estimating the slope and the intercept for the line. So like I said, if we get a new sample from our organisms, we don't even need to see it. If you just give me the measurements of length and weight, I can plot it in this plane. And if it falls in the lizard region, I say it's a lizard. If it's fall on the frog region, it is a frog. The important key point here is that I'm not looking at the animal anymore. All I care about is these two measurements. And finally, if I want to put this information into the computer, I just need to program it. And that's where computer science comes into play. So if I just a simple program in which if the weight is greater than the line, so I'm on this region up here, I will say that this animal is a frog. If it is below the line, I will say that it is a lizard. So I will put all these things together, and now my computer will be able to identify frogs from lizards. And these are the three main components, like I said. And of course, you might be thinking at this point, well, really, a frog is not just the length and the weight, right? The frog has a lot of other features and characteristics that are not fully explained by length and weight. And you're right. We are making a very simplistic model in which we assume that the frog is just the combination of its length and weight. We could actually add other features for the frog or for the animals. We can add the color so that we will be living in a three-dimensional space instead of two. And now the idea would be to find planes that separate frogs from lizards. And we don't need to stop at three. We can actually keep adding all the features that we want. What do they eat? Do they have fins or not? Do they have eggs or not? Are they mammal or not? So we really can measure everything that we want from the organisms, and that becomes this vector of measurements. And as you see, the last one, we can also sequence gene or genetic information from the organisms, and that can be something that we also add. So mathematically, we're just converting the physical frog into this vector. Why? Because this vector is something that the computer can read and can understand. So the more that we measure, the higher the dimension space where we will be living. So of course, that has certain complications that I will talk about later. So this is what we call a model assumption. We are assuming that we are measuring everything that makes a frog unique, all the characteristics that make a frog be a frog. And of course, this will always be simplistic. Mathematics will always be a simple version of reality. So we will never be tackling the entire frog. And but what I want to highlight here, me coming from math, is that we don't need to. we don't really need to have everything that the frog is in order for us to have models to distinguish, in this case, frogs from lizards. And you will see one example. Well, actually, before, there is this very famous quote by George Box. He is the founder of the statistics department here at UW-Madison. And he said, all models are wrong, but some models are useful. And that's the important thing. Like by saying all models are wrong, that means we will never capture the entire frog in this mathematical simplification. But if we choose these features adequately, we will capture what we need from the frog in order for us to distinguish what we want to. And I'm going to show you one example just to convince you that we don't really need to measure everything to have a useful model. So this is a very realistic map of a subway. And I think this is Massachusetts, I'm not even sure. This is very realistic. So this is a photo taken from satellite. So this is exactly how it looks. And if we contrast that, this is a very practical map of the subway. This is not done like the length is not up to, like, it's measured exactly as the reality is. Everything is not exactly as it should be geographically. But when we go to the subway, we of course find the one on the left more useful if we want to navigate the subway, right? So our model, it doesn't have to capture reality exactly for it to be useful. It only has to be done in a smart way so that it captures the features that we need for the decisions that we want to make. So going back to our example, yes, all models are wrong, but some models are useful, and that's the important thing. And now you might, I mean, I'm sure you all can guess that distinguishing frogs from lizards is not really my job, but I wanted to start with this example, and then I can get into the things that I do as a statistician. And one of the examples that I work with, and actually Tom was telling that you will hear about this in February, is this relation of human health and microbiome, specifically gut microbiome, because we have microbiome in the lung, in the skin, there's in many different places, but in particular, the microbiome in the gut has been linked to different human diseases, diabetes, cardiovascular disease, Alzheimer even. So what I do as a statistician is try to relate now the communities that live in our gut and whether they play a role into the development of these certain diseases. So this is exactly the same framework as the frog lizard example, and you will see, because we have a bunch of humans that we collect from our samples. Some are sick with some disease, let's say diabetes. Some are healthy individuals, so we need to collect both. And now what we really want is we want to categorize and classify the groups that are sick and the groups that are healthy. But in this case, this is harder because with the frogs and the lizards, we can do that by eye, right? But in this case, how do we know? Like, we cannot even do it by looking at the microbiome data, right? So of course, there's a more complication about how we make this distinction, but in the core, the problem is similar. We want to classify what is it about healthy individuals that make them healthy? Is there something about the gut microbiome, the characteristics of the microbiome that is similar or is shared across all healthy individuals? And similarly, is there something about the microbiome on the sick individuals that we can think is related to the presence of the disease? So if we remember again, the frog example, if we want to make this classification, we need to first find a way to represent the microbial communities in a mathematical way. You remember we had the frog, but we represented that as a list of measurements that we can plot in a mathematical space. So we need that for the microbial communities. And the second thing, we need to know, how do we classify sick from healthy? If we go back to the frogs and lizards, we had this plane and a line that separated frogs and lizard space. So just thinking about the frogs, we had this mathematical vector and the line that classified them. We need the exact same tools, but now for the microbial communities. And here's where you see that it's not as simple as just a mathematical vector anymore. So I will show you just two examples. One of them is very related to what I do. We represent the microbial communities in what we call phylogenetic networks. A phylogenetic network is a graph that represents the evolutionary relationship between different communities. So if you are familiar with the tree of life, if you have seen talks about evolutionary biology, it is a similar idea to a phylogenetic tree, but phylogenetic networks allow for hybridization between different communities. So the way that we represent the microbial community of someone's gut would be with one of these networks. This is our mathematical object. And then we have a bunch of networks, one for each patient. And now we want to classify, identify which one corresponds to sick, which one corresponds to healthy. And one way in which we do this, and this is also, it's not that straightforward. So I'm skipping some details under the rug, but we project these networks into a high dimensional space that is called BHB space. And all that you need to know for now is that each of these networks becomes a point in the space. So that again, it's similar to what we did with the frog. We have a cloud of points and we can see, oh, actually there is a pattern. All the ones that are sick are up here, all the ones that are healthy are down here. So ultimately you will see that data science is just transforming what's in real life into a space where we can start to look for patterns. There are a lot of challenges in this, and of course, I don't want to get into many of the messy details. So let me just skip that for now. And I will actually talk a little bit more about the phylogenetic networks because that's the bulk of the work that I do, that I'm currently doing. But there's a second way, there's a different way in which we can represent the microbial communities, and that is in terms of matrix. So this is a matrix in which each row corresponds to one individual, one gut, and then columns correspond to different communities. So you will see whether a certain community is present in that gut or no. So it's like a 0-1 matrix. You put a number 1 here in that cell if there is if that community is present in that gut, or a 0 if it's not present there. So then again, the microbial community is represented, is fully represented by this matrix, presence-absence matrix. And then we have this collection of matrices, so we can, again, distinguish sick from healthy by doing standard tools like regression or more machine learning type of tools like neural networks. And I'm gonna focus mostly on, like I said, the phylogenetic networks part, but if there's interest from people, I can also talk about the second approach at the end of the talk. Okay, so yeah, this is what I'm gonna be focusing on. So just very briefly, let me check how much, oh yeah, okay, the time. So I'm just gonna focus on phylogenetic networks and I'm gonna guide you through 4 main questions I wanna try to answer. What are they? Why do we need to care about networks, and how do we get them in practice, and when do we need to get them? And actually, in fact, I might have caught some of those questions for the sake of time. So I'm sorry if I'm lying to you right now. But anyway, but I can answer the questions at the end if you want to know if I skip those. So I mentioned very briefly, what are phylogenetic networks? This is a graph that is representing evolutionary relationships between certain organisms. So for example, here, what we have is, well, I changed the species. Now these are fish, but it's just because these tools can actually be applied to different organisms. So it could be bacteria, it can be fish, it can be plants. And that's one of the cool things also being a statistician, is that I get to collaborate with people from a variety of organisms. I've been working with, this type of fish, and then carrots and potatoes, and of course plants, and well, anyway, a variety of things. And the tools, the mathematical tools, are the same. You need to tailor them, of course, to different organisms, but in the core, they remain very similar. So I changed the example, and now we have fish. But here, the idea is the same. We have a network that represents how closely related they are to each other. And here there is a time progression from left to right, so that if we move towards the left of the tree, we are going back in time to ancestor species. So the one here, we usually don't draw the nodes, but I'm circling, I'm hoping you can see my cursor. So the very last line at the left, that corresponds to the root of the network, and that represents the ancestral species that is the ancestor of all these 4 species of fish. And then as time progresses, the ancestor of all 4 diverged into two different species just by random mutation and by the evolutionary process, and it became two different species. So one went up here and the other one went down here, that eventually evolved into the fish at the very bottom. So at each of these intersections, there is an internal node that we don't draw, but there's an internal node that represents the ancestor of that clade, all the clade that goes to the right. The difference between a standard phylogenetic tree, for people who have seen this before, and a network is that we have these extra arrows, like represented here in green, that they correspond to any form of gene flow. So it could be hybridization, it could be horizontal gene transfer, introgression. There are many biological processes that cause this transfer of genes from one population to another. And of course, this is more common in certain organisms than others. For example, these fish, they hybridize a lot. So they all live in the same river and they all create different hybrid species. So sometimes it's hard to tell which species each fish correspond to. Bacteria as well, they do a lot of gene transfer, even if they are different species. Well, I mean, I don't want to get into the concept of species from bacteria, but even if they are different species, they can still create, share different genes, and that's represented through this arrow. So yeah, the phylogenetic tree is the one when we don't have the arrow. It's a fully bifurcating graph, where if two lineages, if two edges diverge, they never join back again. And oh, I forgot that I had the nodes. I should have waited until this moment. So yeah, the orange nodes are the internal nodes that represent ancestral species, and the green nodes represent living species for which we have data. We sample data from these species. And one of the characteristics of phylogenetics is that we do not have data for all the species. So for example, we can only collect data for the fish that we can collect. We go into the field, we grab them, we sequence them. But the ancestral species, the one that lived millions of years ago or however long ago, we don't observe that. We don't have DNA sequence for them. So we don't know what was the genetic sequence for those ancestral species. So this is a very interesting pattern of missingness in the data. You might have heard, when you have data and you have missingness, you need to account for that, that you don't have all the information that you might need. And in this particular case, you never, or I want to say never or rarely have the data for the ancestral species in the tree. And when I keep saying data, and what I mean by data is genetic sequences, and the genetic sequences are simply represented as a string of the 4 nucleotides. So we know that the DNA is comprised of 4 nucleotides, ACGT. So when we sequence a specific gene, this is simply a string of these 4 nucleotides. So it's a combination of ACGT. In this case, I'm showing a toy example. Obviously, it's a very short gene of, I think, 8, 9 nucleotides, however number, I cannot count. This is a toy gene, of course, but in reality, we have genes that are like hundreds of thousands of nucleotides long. So what happens is that we have these sequences. We don't know the tree. This is what we want to estimate. We don't know what it is. And the information that we have, our data, are simply these sequences. So we want to go from the sequences that we collected from the organisms. And again, I said, I changed the example, and this is fish, but the sequences could come from bacteria as well, so the bacteria in our gut. So we sequence these genetic sequences, and then we want to estimate the tree from the sequences. And this is what we call phylogenetic inference. So the first step to estimate the tree is actually to guess it. And that doesn't sound very scientific. I'm sure you're all like thinking, okay, what is happening right now? The phylogenetic inference process starts by us guessing the starting tree, guessing what could be the possible tree. So let me go back one slide. So we have our sequences, we don't know the tree, and we're gonna start first by guessing one tree that represents the sequences. This is the first step. And the second step is we want to calculate the likelihood of this tree. And by likelihood, we mean how well this tree explains the data that we observe. So the likelihood is a measure of how well this tree is representing or is in alignment with the data that we collected. If a tree has a very high likelihood, that means that it is a tree that agrees with the data. So it's a good tree, it's a possible tree. If a tree has a very low likelihood, that means that it is not in agreement with the data, so it's probably not the tree that represents the evolutionary relationships. And I wanted to show just these parentheses a little bit about likelihood, because I mean, I don't know if people are familiar with the likelihood function, but it's just any function where our parameter of interest, we usually in math like to call it theta, but this it could be the tree. And our goal is to find which is the value of our parameter that maximizes the likelihood. So now this is an optimization problem. And we start, like I said, we start in a random place, we evaluate the likelihood, and then we move to another random place, evaluate the likelihood, move to another random place, evaluate the likelihood, and you see where I'm going. So we are guessing different trees and evaluating the likelihood of all these trees. And at one point, we evaluate all the trees that we have, and then we collect or we keep the one that has the highest likelihood. And in our case, like I said, in mathematics, we call this parameter theta, but really what we have are trees. So we start with one tree, we evaluate the likelihood, move to a different tree, evaluate the likelihood, and then we're jumping in this tree space until we find the one that maximizes the likelihood. And again, to repeat, the likelihood is a function that is quantifying whether that tree agrees with the data or not. That's why we want to maximize it. Okay, and of course the challenge is that we don't observe this function. We don't know how this function looks like. So if you see, we are blindly searching this space. So we start with a tree, we evaluate, and we're guessing the shape of this function without actually seeing it. So you can think at this point of several weaknesses of our approach. And one of them is very obvious, and that is we need to sample enough trees from our space in order to have a good idea of how the likelihood function looks like. If I only pick 2 trees and I say, oh, this is the maximum, well, but you probably didn't cover the entire tree space to have a good idea of how the likelihood looks, right? So this is just like the recipe that I mentioned of how we find this maximum likelihood. We start in a random place, we evaluate the likelihood, jump to a new tree, evaluate the likelihood. If it is higher, we keep the highest, drop the other one, and we repeat. So we keep searching over and over and over. And the only thing that I have not told you up until this point is how do we calculate the likelihood of a tree? I only told you we calculate the likelihood and this is how we choose, but I have not told you this. And I will tell you that very briefly, because like I said, this is gonna be an hour-long talk just on the likelihood. And I wanted to cover different things that I work on. So yeah, so to calculate the likelihood, we actually don't take the whole gene at the same time. We focus on each side independently. So we just pick one of them, one column. And like I said, we don't observe the state for the internal nodes. So those are all knowns. So we do observe G, G, C, T. Those are the nucleotides that we observe for that position. We do not know what was the nucleotide for the ancestral species, either the X, the Y, and the Z. We don't observe that. And the other thing that we don't observe is the time. So we know that, for example, whatever nucleotide was here in X, it had some time to mutate to the present into the observed G, what we observed finally. And we did know this time by T1. And there's also another time, it doesn't have to be the same time, that it takes to evolve to G2. And with these parameters, we can write the likelihood. And this is the only formula that I have in my slides. But I wanted just to illustrate it. This is the actual formula for the likelihood of this tree, that it's summing over all possible values of X, Y, Z, because we don't care about them. We don't observe them, but they are not the parameter that we want to estimate. So we calculate the likelihood for all these 4 possible options. It could be A, C, G, or T, we calculate them for all of them, and we average across. And then in the core of the likelihood, we just have a product of probabilities. So we have a probability for whatever was the first nucleotide here. It could be randomly chosen between the 4 options. And then we have a bunch of products of transition probabilities. The probability of mutating from Z to T in time T6. This is the first probability here. And then so on and so forth. So I'm just calculating how likely it is for me to mutate from this nucleotide into this other nucleotide. So if Z, for example, was a T, then we want them not to mutate in time T6, just to remain a T. And these mutations, they are all governed by a rate matrix, how likely it is certain mutations to happen. Some models assume, and again, I use the word model, we have all these different types of assumptions. Sometimes we assume that all mutations are equally likely. It is equally likely to jump from A to C to G to T, they're all equally likely. Or for certain organisms, certain times, we can assume that they're not equally likely, that there are some mutations that are more likely than others. So everything is governed by this probability, by this rate matrix. So yeah, these are different types of models, different levels of assumptions, they have different names. So if you see, for a given tree, actually, let me go back. Oh no, I cannot go back. Yes, okay. So for that fixed tree, with this formula, I can calculate the likelihood of this tree. If I give you a different tree, you will have a different formula, but it's built in a similar manner, and you can evaluate the likelihood for that different tree. And again, going back to how do we find the tree that best explain the sequences, we start in a random tree, evaluate the likelihood, jump, evaluate the likelihood, jump, 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 jump. So we are searching the space of trees until we find the one that is in maximum agreement with the data. And yeah, so as I said before, you might think that, well, this seems a bit tricky because if I don't cover the entire space of trees, how can I know that I really found the maximum? So imagine that I only sample these 3 trees. I only choose one, choose another, choose 3, and then I said, oh, this guy is the maximum, right? That's the highest value that I got. So this has to be the maximum. Well, it's not really the maximum because you didn't cover all this part of the tree space. So in order for us to find the true maximum, we really need to cover tree space as best as we can. And that is challenging because the space of trees really just grows out of control to the point where if you have 52 species in your sample, there are more trees than atoms in the universe. So if you imagine, I want to estimate the tree for 52 species, you would have to search the space that has more trees than atoms in the universe. And for each of these trees, you have to evaluate and optimize the likelihood. So obviously this cannot happen, right? You cannot do this. It's just impossible. You, not even with all the computers in the world, I think you could do this gigantic optimization. So what we have to do is we have to find some heuristic algorithms that we hope, and I say this is not, again, not very scientific, but we hope that are going to converge to the maximum likelihood. And there are theoretical guarantees in many cases, but in other cases, we still don't have them. We're still exploring them. And that's part of the job that I do and many other mathematicians. We're trying to find algorithms that will give you some form of certainty or some theoretical guarantees that if you're estimating a tree, then this is in fact the maximum likelihood tree. The other challenge with this, like I said, we really don't know how this function looks like. And how this function looks like depends on the assumptions that we make. So if I assume that this is a GTR model, and I didn't pay much attention to this, but the name of the model depends on what assumptions we make on the rate matrix. So if we assume that this is a GTR model, then our likelihood function will look like this. But if we make a different assumption of the model, the function will be completely different. So the assumption that we make on the model has an impact on our likelihood function, and we might end up finding the maximum, yes, but for the wrong function. So yeah, and of course, we don't know which one is the real function. So we also need tools that help us find which is the function or which is the model that that we have to choose, and that's in more agreement with the data that we observe or the biological process that we're studying. And lastly, the other complication is that the likelihood function also depends on the size of our data. So if we have a gene that has 1,500 sites, our likelihood function will look like this. But if we have fewer sites, our likelihood function will be different, because our function, the likelihood function, is converging to the truth as we accumulate more data. So if our data is not big enough, we can find the maximum, but perhaps it's a different maximum because our likelihood function was not as good because we did not have enough information, because we did not have a big enough sample size. So all of this is to say, I hope this is not super pessimistic, but this is just to say that phylogenetic inference is challenging, and there's a lot of work from computer scientists, mathematicians, statisticians into developing tools that can allow biologists to estimate the tree of life or network in a way that that we can have some confidence on the estimation. So anyway, so this is the process again from the sequences to the tree. So there are many different things that go into this process. Choosing the sequence correctly, have a good sample size, selecting the correct model, like I said, hoping or having algorithms that do converge to the maximum, and ultimately getting a tree for which we want to have some measure of confidence, how confident we are that this is the tree that best is aligned with the data that we observe. Okay. How much time do I have? Okay, so the second question, so it's not going to be all the 4 questions. I think I cut some of them by the end, but one of the things that I want to highlight is that people that are reconstructing phylogenetic trees, many times they don't care about networks. They say, you know, I don't really care about those extra arrows. I just want the main signal of my data, the main bifurcation process of my data, which is, I mean, it's great, but there is the problem that if those gene flow events were part of the history and we ignore them, then we might end up with the wrong tree. That is the danger, that we might end up, and we did this study we published in 2016, that if you use a tree method and there was a gene flow event, you could reconstruct the wrong tree with a very high probability. So this is just a simulation study that we did in which we knew that the data came from a network here on the top right corner. So we knew the data came from a network, but we pretended we didn't know, and we used a tree method, a method that ignores the gene flow event. And what we saw is that, let me just focus on the bottom row. Here in white is the proportion of the times that we estimated the correct tree. So that's the tree without the blue arrow, so the black tree. You will see that as I'm increasing my sample size in the x-axis, so that's the number of genes, this white bar is really converging to 0. So this means that as I accumulate more data, I'm converging to the wrong tree with very high support. So the wrong tree is very highly supported by the data. And that's scary because we tend to believe that as we accumulate more data, we will converge to the truth. But it turns out that this, again, is one of those model violations, the assumption violations. If you have a network, if the evolution of your organisms is better explained with a network, and you force a tree to be estimated, then you will end up with the wrong tree. So here, this is to highlight again the choice that we're making. So basically, what we're saying is that we can estimate the wrong tree if we ignore that gene flow event. And then, oh gosh, I'm okay. Then apparently I am talking about the 4 questions. Okay, so how do we estimate a network? The thing that we do is that we have the whole genome. So the whole genome is represented by these gray bars. And for different genes in the genome, we estimate what we call a gene tree. And we do that with the maximum likelihood process that I just described. So for that specific chunk of the genome, we run our maximum likelihood machinery and we get a tree. And now the question is, we want to summarize the information of all the different gene trees that we get from all the whole genome. into the reconstruction of the evolutionary history of the species. And this species, like I said, we can assume that it is a tree, or we can assume that it is a network. And again, reiterating, this is a model assumption. It's something that we don't know, and it's something that the data will not tell us explicitly. We will make the assumption, yes, this is well explained with a tree, I'm gonna get a tree at the end, or no, actually, I think I need to invoke a network. And there are model selection tools, and that's one way in which we can choose which one to do. But of course, this is a choice of the model that we're making. And for the case of the networks, I want to highlight that when I was doing my PhD here with Cecilia Nesh, she was my PhD advisor in statistics, we created this tool to reconstruct networks that's called SNAC. And this is the one that I was just going to highlight here. SNAC will allow biologists to go from the collection of gene trees as the data into a network that represents the evolutionary relationship of the species involved in the organism. And this is a Julia package. I don't know if people are familiar with the Julia programming language. It is similar to R or Python, but in my experience, it is much faster and I like it better. It's very easy. It's a high-level language, so it's very easy to program in Julia, but it is, and at the same time, it's very efficient as a language compared to R or Python, at least in my experience. So one thing that we cannot, is what we call identifiability issues. And that is, we're trying to find the maximum of some function. We always try to find the maximum. That's the one that has it agrees with the data better, the maximum agreement with data. But what if our function looks like the one on the right? So if our function is completely flat, then there is no maximum, really, right? We cannot find, like, there is no maximum. So whatever we find will be equally well explained, and it will equally explain the data. So this is a completely flat likelihood, means that our parameter, the parameter that we're trying to estimate, is really not identifiable. The data does not give us information about which is the best parameter, in this case, which is the best tree. And when we are creating these methods, we want to make sure that the data that we have really allow us to identify the best parameter. Otherwise, I'm just going to give you one of these trees chosen randomly, and it's not going to really explain, or it's not going to really have the interpretation that you want. So in our case, we did this study for SNAC. We wanted to make sure that if we estimated a network, it was actually the maximum, in our case, was pseudo likelihood network, not just a randomly chosen network. And it turns out that what we found is that the cycles, the hybridization cycles that were very small, 2 nodes or 3 nodes or 4 nodes in the hybridization cycle created in the network, if these cycles are very small, then it was very hard to detect these networks, these cycles. If the cycle was very big, then we can always detect it with very high confidence. And mathematically, this makes sense, and that's cool. But biologically, it doesn't make any sense, because if the cycle is very big, that means that the donor and the recipient of gene flow are very far apart in the tree. And it's very unlikely that they will share any genes or there will be any hybridization if these are very far or distantly related species. It is more common to have gene flow or hybridization between closely related species, which would create smaller cycles in the network. But sadly, those are the cycles that are harder to be detected with the data that we have. So we're still working on new models or better models that will allow us to estimate these cycles that are more realistic or that appear more frequently in biology, but our current methods are not able to estimate accurately. And to show you why this matters, we did some simulations where we have 2 cycles in a network that has 2 cycles, one in orange, one in red. One is called a good diamond and the other is a bad diamond. And it is a diamond because it has 4 nodes in the cycle. And you will see why one is good and the other is bad. Here we have the network, we simulated data, and then we counted how many genes we need to estimate this cycle. And it turns out that to estimate the good diamond, you need as little as 30 genes. So this is where here you have a very high accuracy. This is a triangle for the good diamond. So you have very high accuracy with as little as 30 genes. But if you want to estimate the bad diamond, you need 3,000 genes. And that's because you remember that I told you the likelihood depends on the data size. So the likelihood for the bad diamond is very flat until you get 3,000 genes. That starts to be a bit more picky, like to find the maximum. But the good diamond likelihood is not like that. With as little as 30 genes, it's already kind of pointing at the maximum. So we use this example to highlight that identifiability is important, and many times people that develop these methods, they don't take the time to check whether their method is actually identifying what they want to identify or not. And that is a danger, because the users of these methods, sometimes they just plug in their data, there's a black box, they get a network back or a tree, and it could be just a randomly chosen tree if the method is not finding the correct maximum, or if the likelihood is very flat because the tree is not identifiable in this case. Anyway, so the last question, yeah, we made it through all four. When do we need to when can we get, when do we need to choose to use a network method versus a tree method? And there was this hypothesis of goodness of fit test that was done in the lab with Cecile and where basically it's just a test whether a tree is a good enough fit for my data. So if my data is well explained with a tree, I stop there. I don't need to go get a more complex model, which is a network. If it's not a good enough explanation for my data, then I choose a network. And we use our tool SNAC into this data set from the pretty fish that are 24 species of swordtails and platyfish. And the swordtails, they have this sword on the tail, and the platyfish, they don't have the sword in the tail. And our method was one of the first methods that could tackle a data set with 24 species. 24 doesn't even sound that big, but existing network methods could not handle more than 10 species. So ours was one of the first that was able to increase to the gigantic size of 24 species. And it's just that network reconstruction is very challenging. And as you can probably guess by now, it's just because the network space is just so big. And as you increase the number of species, it's growing out of control. So we don't have guarantees that we're finding the maximum likelihood or whatever function we are optimizing. But anyway, so we reconstructed the network, and we actually found gene flow events that were in agreement with what the original study had found. Okay, so that was the short story, or really a relatively long story, of what I do and how we get a phylogenetic network. And I apologize that I changed the species in the middle, but I have not done one such a big network for bacteria. So I only have the example with the fish. But these would be the exact same tools if we want to estimate the network for the microbiome, because the difference is that instead of different species of fish, we would have different microbial communities, but the algorithm would be the same. We would be estimating the gene trees, we would be estimating the network. And in particular, one thing that I'm working on right now, and this is ongoing project, so I don't have any results yet, is precisely to tailor our methods to the particular case of microbial communities. And the challenge there, like I said, mathematically, the core, the method is similar and doesn't care if you have fish, if you have plants, if you have bacteria, it doesn't care. But the models that we're going to be choosing to represent this evolution, they do need to be tailored because they don't evolve the same. Bacteria doesn't evolve the same as fish, right? And the other complication is that when we collect microbiome data, that's also done in a different way than when we collect data from fish. Because when we collect the data from fish, we grab the fish and then we sequence it, and we know that this sequence came from this fish. But when we do microbiome, we pretty much either gut or soil, because I also work with soil microbiome. We grab, let's just say soil, piece of soil, and then we sequence everything that's there. So we don't know which sequence corresponds to which bacteria or to which community, microbial community. So we don't have that correspondence anymore. And all of our methods rely on us having not just perfectly aligned sequences, but also to know which sequence corresponds to which species or to which individual. And we no longer have that for microbiome. So for microbiome, we just have a bunch of reads that we sequence. So we grab the soil, we sequence everything. We don't know who belongs to whom. So we need to first cluster the reads into the different species of the different groups. And then from each group, we need to align them. And then we start the process of the network. So there's an extra step for microbiome data that I'm still working on. This is an ongoing process. And of course, just like anything, statistics, like when you have all these different steps in your pipeline, every step can have estimation errors. So the very first step has estimation errors and is just propagated through this whole pipeline. So it's a question for us statisticians, how do we handle this propagation of errors, and how do we incorporate that information into the next step? Okay, so just a summary, many challenges in phylogenetic networks or in general, trees as well, identifiability, scalability, how do we get networks from metagenome? And the very last topic that I wanted to show, oh, actually I have less than 5 minutes, I'm sorry. It's just one that I find it really interesting, but all this talk that I've been telling you is that we have samples of gene trees. The gene trees are our data. And if we had numbers, just pretend for a moment, if you have a sample of numbers, we've all had to work with numbers. We know what to do with them. So we can do histograms, we can calculate the mean, the standard deviation, we can do box plots. We know how to work with numbers. But now we don't have numbers, we have trees. And the trees, some very smart people created this space where, as I told you before, each tree can be represented by just one point in this space. So you forget that they were a tree, you just treat them as a number. So it is very interesting that there is such a space that for such a tree space where we can calculate the average of the tree or the variance of a collection of trees. So I just wanted to point this out as something that I find really exciting, that there's a lot of work, not just from the statistics part, of the optimization part, of the computer science part, but also from the mathematical part, where we come up with these weird spaces and do statistics on them. So that was the second part of here. Okay, so that's the end. Thank you for, I'm sorry that I was planning for 45 minutes so that we have more time for questions. This is just the acknowledgements at the end for the students that are working with me and my collaborators. And I'm happy to take any questions that you might have.
Thank you very much, Claudia. Does anybody, I'm gonna look in the chat, see what we got here. Can you read Claudia Rajgopal's question? I'll read it for everybody. In the tree identification algorithm, do you also consider the lifespan of an individual organism? Also, can you predict the survival or extinction of the tree peering into the future? Does Ancestry.com use any similar approach?
Okay, so there are many questions, I think. So I forgot the first one. Let me answer the second one, which is we can predict. And I think I don't work on that. I have not done any of these, but I do know that people that work on infectious diseases or trees or networks related to infectious diseases. So people, for example, that are working with COVID or like tuberculosis or different viruses, they do try to predict or to get ahead of that evolution. Whether that's successful or not, again, it depends on many things, the method they're using, the models, but I do know that there is some theory on that. I just don't have any experience on it.
And then the first part of this question, if you don't mind, I'll reread that because that's the one I think.
Thank you.
It says, in the tree identification algorithm, do you also consider the lifespan of individual organisms?
We don't. The methods that I described here doesn't include that information, but it's something that it could be included. I guess I don't know very well how, but I mean, it's just like all these models, they are tailored to different information, so we can always include that. But the current method that I described does not.
Good. And the ancestry.com angle you've already covered?
No, I haven't. So the Ancestry, probably they do something similar. I don't think they try to get the entire tree. I think they do more like sequence similarity. So they have they get your sequence, and then they have a database of sequences from different places, different people, and then they just try to find which is the sequence that is most close to your sequence. So I don't think they try to get the whole tree. They just try to find which are you closest to in a database.
Very good.
But I don't know very much, so don't quote me on that. That's what I think they do.
Great. And then if you want to open up the chat, I'll read it
so
everybody can hear it, but you can also read it.
Okay.
Ryan asks, I hear a podcast that graphed words in different languages into a three-dimensional space. And it showed that language structure is similar across languages in this three-dimensional space. Is this the same as BHV that you know of?
It's not the same, but it has very similar ideas. And yes, I also saw that about language. They are embedding this language into these vector spaces to do the natural language processing. And it's very interesting, all the patterns that they find. The BHV space is actually built on not the sequences, the branch lengths. So the way you embed them is with branch lengths, but there are different spaces that we could use. And one thing that I've seen is that people are embedding sequences into different spaces, and sequences are more similar to language. So I think they will find similar things when they embed sequences, not the trees. But yeah, I mean, I think it's very, very interesting that you can find these patterns from these weird objects.
Great. And then Ryan adds, and they're looking at animal and looking at animal communication now.
Oh no, I have not seen that. Wow. Yeah. Okay.
For those of us who remember Flipper back in the 60s, that'll be great. Diana asks, when you read the general literature, how often do you see suspect phylogenetic trees? In other words, how good are most phylogenetic trees? I can think of some major reshuffles.
Yeah, this is a great question. And sometimes I am suspicious. So the first thing that I read when I read literature is just the methods part, which is usually the part that they tend to hide after the appendix, and they have like smaller font even. That's the first thing that I read. And that's when I know whether I trust or not that much the tree, because like I said, I don't have enough, I'm not a biologist, so I don't have enough knowledge on the organisms to say, this doesn't look right. But I do know about the method, so I can read that part and say like, it doesn't sound like it was done properly. And I cannot say, okay, this proportion that I see, I don't trust, but I have seen some where I'm suspicious about the estimation process. So, but I think in general, people do a good job, but I have found some instances where they are not ideal.
I'm going to ask some questions because I don't know how this works. You're dealing with a huge amount of data. I'm assuming it's sequence data. I'm assuming it's DNA sequence data, although you could probably do amino acid or other linear sequences. Where do these databases reside? Who gets access to them? And what do you do when the data gets improved or cleaned?
Yeah, no, this is a great question. So there are many databases, for example, like BLAST, I think is one of the most widely used. There are these public databases where people, so there are different ways. So specific labs collect their own data, like they go to the field, they collect their samples, they sequence them, they store them in the computer, and then when they're publishing some paper or something, some journals require that you make the sequences publicly available. So then you put them in this online database that is public and people can use it. Sometimes people don't do a great job putting clean sequences. They can be like really raw. But in general, there are a lot sequences. And of course, there are organisms that are more studied than others. So there are perhaps a lot of sequences for Arabidopsis, right? But there are like very few sequences for something else that I don't even know. So the proportions of publicly available sequences is not uniformly at random across the tree of life. But there are these databases. And if they have cleaner versions, I mean, they're expected to update them, but I don't know if everybody does that. So
And then,
did that answer your question, Tom?
yeah, that's a very good start. And then Larry Winkler asks, is BHV validated against morphological determined species?
That's a great question. And I don't think it has been done, but it would be interesting to do. So right now, people are just starting to explore. So BHV has been just mathematical space for a while. It is very recent that we're starting to see whether the average tree or the patterns that we see have actual meaning in biology. So I think there's a lot of work that will still be working on BHV space. But to answer your question, no, not yet.
Other questions from folks? If you want to unmute and speak your question, or you can type it into the chat. I'm checking the YouTube and there's nothing new on the chat there. I'd like to pursue the amino acid versus DNA sequencing. Is there any advantage? Are there any disadvantages to doing amino acid sequences rather than DNA sequences, given the nature of the degeneracy of the translation code?
Yeah, no, this is a great question. And actually you put me a bit in the spot because I don't know. There are models, so I work with both and they all have their respective models. So you have the model for evolution of the amino acids, you have the model for the evolution of the DNA. I have never done a comparison to assess if one is better than the other. I mean, I've just used the available data that I have, and many times you only have the amino acid sequences, many times you only have the DNA. There might be people who have done this comparison, but I have not done it, so I wouldn't know if there's one better than the other.
I'm just wondering, when we do our little intros here with DNA, often I point out to kids that the way, I ask them, how do the Canadians spell color? Or how do the Canadians spell center? And that there are minor differences. When you hear the word, that's kind of like the amino acid, you can't tell that there's a difference, but when you see it's spelled out, it's more like the DNA. So I'm wondering if that's just my little etymological tendencies coming through, and I have no idea if it fits with DNA and amino acids.
I mean, there might be. Yeah. I mean, there could be. Yeah, I'm sure there is a difference. I wouldn't necessarily think that it would be always one better than the other. I think there would be different cases when one is better than the other and the other way around. But I wouldn't know.
Are there any other questions? Because I got one or two more, and I'd rather hear what people would like to ask. But I'll go ahead and ask this one then. Gene flow is a big deal in nature and recombinant DNA technology. Gene regulation, you mentioned that a couple times, horizontal versus vertical. How big does a DNA sequence flowing to make a network? Is it a single gene? Does it have to be a gene cluster? How tiny, how large does a horizontal gene flow have to be for you to pick it up with your method?
Yeah, no, this is a great question. And we cannot detect it if we have only one gene.
Okay.
Because of the model, the method that we have, as I mentioned briefly, we first reconstruct trees for different genes, and then the signal from gene flow, we get it from the differences of the gene trees. So we need to have a lot of gene trees so that we are able to detect, okay, what is really vertical signal that is tree-like, what looks like doesn't fit the tree that could be better explained with a gene flow event. So we need many genes as many as possible. And in fact, the larger the network, the more genes that we need. So we were doing some studies that for certain networks of certain size, for example, if you want to estimate a network for more than 20 species, you need in the order of thousands of genes. And the other thing is you need longish genes, like you don't, like each of these blocks to estimate the gene tree, it has to be, like again, people as a statistician, they always ask me for numbers of sample sizes, and I never want to commit, especially if I'm being recorded, but you want a sufficiently large gene that will give you an accurate gene tree. So you need many genes and with sufficient number of nucleotides, each of these blocks. And I should also clarify one thing. I've been saying genes, 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 but they don't have to be protein coding genes necessarily.
Yeah.
So people have called them genes, but actually they have this new term called coalescent gene. It's just a block. So it can be introns, it can be non-coding regions. It's just a chunk of the genome that the only assumption, I should have said this, is that there is no recombination within this region.
Okay.
That is the definition of our coalescent gene.
Other questions? Thank you, Claudia. Other questions? So one last one I wouldn't mind knowing. What's the name of the math classes that a student takes to learn how to do this stuff?
Wow, that's a very good question. And I took linear algebra. I guess what level? So we're talking college or
Whatever
high school?
it takes to, I have no idea.
Okay, so let's just say college. So I think linear algebra is very important. Calculus, obviously, probability is important as well, and something of optimization where they show you how do we find the maximum for functions that we just don't know and don't observe.
Okay.
There are those classes. Yeah, at least those are like the core.
Are there math classes where you learn how to do these phylogenetic trees, that type of thing?
No,
Or
so I didn't
pick this up as a grad student.
I picked that up as a grad student, yes. But to be honest, I also didn't search for them. I did not have an interest in biology when I was a college student. I wanted to do just pure math. The interest in biology came when I was a grad student. So perhaps there are, but I just didn't search for them when I was in college.
So Ryan is pointing out that Ryan took college calculus 4 years ago and followed the lecture, but didn't understand the actual math, if that makes sense. And
yeah,
I didn't take calculus. Calculus took me, and it was a lot more than 4 years ago. So that's why I was interested in what kind of classes you would take, or if this is the kind of thing that you pick up as a grad student in labs, or just pretty cool stuff.
Yeah, so I think I had a good core of math, linear algebra, and calculus and probability, but then all the trees thing that was completely in grad school.
Great.
Yeah.
Okay, well, thank you very much. If there's
I think there was one last question. Sorry, Tom. I think there was one in the chat. Are the trees or networks randomly generated, or is there some assumption or knowledge that suggests the next tree? When we're searching, I am assuming when we're searching for them, they're not completely at random, but they are not. There's no biological knowledge that we use to search. So we start somewhere and then we modify the existing one to get another one and another one. So it's kind of like a chain, but it's not. We like to use knowledge for the very first one. So I said that we guess it, but then as the space grows, we actually need to guess it, do an educated guess. So we use biology to inform the very first one. But then from there, we just walk along a chain of changes.
Great, and thank you for noting Larry's question. I focused on Ryan's that came in after his. Oh, thank you for asking Larry. Dareth, you popped in. What do you think?
It's beyond my math ability.
Math ability?
I'm another one that struggled through calculus and basically quit using higher math at that point.
That's very, I think the way that it's graphically represented is pretty cool. I can't say I follow it, but I learned a lot from the talk today, and I appreciate it. Well, thank you, Claudia, and thank you everybody, and then look forward to seeing you next week for Sean Carroll's talk on his most current book. talk with you soon.
Thank
And
you very
thanks again,
much.
Claudia. This was great.
Thanks,
Thank you.
Claudia.
again.
Thank you.
Thank you.
There we are. Yeah. Wow, Dareth, I'm with you on the calculus, man. I took a second semester of calculus. I didn't have to, but I was doing it for grins and giggles. Oh.
Grins and giggles. Uh-huh.
Well, somebody said you master the math of the second to last class that you take. And I wanted to make sure I got down the first semester of calculus because it seemed to be so important for so much stuff. So I took the second semester, and I think it probably helped me solidify my first semester stuff, but I don't know. And that's one of the things.
I feel like sometimes professors or teachers, they don't make enough connection to reality when they teach math. So I feel like that sometimes that's lost. Like, and that was my experience as well when I was learning math. And I felt like, oh, if they made more connections, perhaps people will continue to be interested in the topics. But
I know my kids have the advantage, even though they're in 8th grade and a sophomore in high school, they can go find YouTube videos that cover math now in a way that all I could do is go look for another math textbook. And you know, you don't have math textbooks laying around, but I hope that makes it easier for kids today to be able to pick up math because they've got the Khan Academy and a whole bunch of other things that they can look to.
Yes, there are a lot of resources for sure.
Well, I appreciate this all, and
thank you.
How are you doing, Jay?
Yeah, I'm doing fine. How are you?
I'm wonderful. You look like you have a red lamp going on in your room.
Yeah, well, it won't be red in a few seconds.
It changes. It's one of those.
Yeah, it's a lamp that changes colors.
Oh.
It's for decorative purposes.
Very good.
Now it's blue.
All right. Well, I'm going to let Claudia go. I'm going to end everything, and thank you very much for hanging out.
Sure.
A little bit of lingering at the lectern here, Claudia, and really appreciate your patience of being delayed 10 months and still hanging
out. No. Yeah, I mean, I remember when we were talking about that day.
Oh,
we were like, oh, yeah.
It was like, because I think it was until 3 in the afternoon or so that we had to pull the plug, and it was like,
yes, it was. Yeah, I remember it was very close. Like I was ready. We were ready.
Yeah.
But anyway, here we are. So it's good.
Zoom. Yes. All right.
Yeah.
Thank you all very much.
Thank you.
See you.
Bye.